think maybe we can slowly start, if that's okay.
Okay.
So wonderful. So first of all, welcome everyone to the webinar of Longevity Education Hub. We are a platform with courses of healthy longevity medicine for physicians, scientists, healthcare professionals, and pretty much for everybody. But at least it's made for physicians in a language that we clinicians, are used to. And we are voluntary based. One of the things that was developed along the now 4 years that we are together as a group was this series of webinars. And we are so extremely grateful for your time, Dr. Herzog, for your wisdom that you will share today with us, and really for being here and to speak about your research that is so crucial for the field. But allow me also just to mention, I think, very, very few of your achievements. So Dr. Herzog is, of course, extremely accomplished. She has a master from Innsbruck on molecular medicine, and then also PhD in neuroscience in Edinburgh. But in between, in her free time, she also acts as one of the executive board members of the Biomarkers of Aging Consortium at Harvard, and organizing the Biomarkers of Aging Consortium conference, and is also engaging in a lot of activities at the King's College, since she's a King's Prize fellow, in terms of education for scientists and for broader audience. And she's focusing in her research on epigenetics and how it is involved in the processes of aging. How can we actually intervene through that path to change those trajectories? Chiara, thank you so very much. We are honored, and the floor is yours.
Thank you so much for the kind invitation. It's a great honor to present to the HLMS seminar, and I've been following your work for a long time. So yeah, pleasure to be here. As Evelyne kindly mentioned, I'm a King's Prize Fellow at King's College London. Essentially, that means I'm starting my own lab there. And I also serve on the executive committee of the Biomarkers of Aging Consortium. Some of the work that I'm presenting today was also done in my former role at the University of Innsbruck, where we've done a lot of work on cancer and cancer detection and epigenetics. So hopefully I can share some interesting insights with you. Great, let's get started. So let me just see if I can move my slides. Yes. So one thing that is the very good news is that over the past decades and centuries, we've really managed to extend human lifespan a lot. So maybe 100 years ago, on average, people would have maybe become only 40 years of age, whereas now we're really much older on average. However, one of the challenges is that this extended lifespan has not necessarily translated to extending health span. So in Austria, where I'm from, the average age of health span is around 57 years of age. However, combined with the fact that the lifespan is around 80 years, we know that we have at least one age-related disease for the past maybe 20, 30 years. And that's maybe a problem because, this comes not only with healthcare burden, but also burden for individuals. Austria is not alone in this. So here's a map of Europe with data on life expectancy. In women, in men, it's similar, although sometimes slightly lower. However, if we look at the same map and color scale and then look at the healthy life expectancy, we see that this is a lot lower throughout many European countries. And this is also true for the large majority of the globe. So when we're looking into aging, what we don't really want to do is extend this period of age-related disease and extend lifespan at all costs. So this would maybe mean more medication, life-prolonging therapies, which maybe don't come with an improvement in quality of life. But what we really want is to push this period of health span and extend healthy life, possibly also, of course, with extending lifespan in general, although opinions become very divided depending on whom you speak to. But this is not the point of the current talk. And one thing that I found very exciting over the years, I come from essentially a background in neuroscience and cancer, and I've been working on different diseases in the past. But one thing that is very exciting is that actually aging is possibly a root cause for many of these diseases. So if we understand the underlying processes, we could delay, reverse, or prevent the onset of multiple age-related diseases in parallel. So maybe we don't need to find a cure for every single disease. And I think this is quite a powerful approach. Um, I'm also very interested in prevention in general, and we know that prevention maybe occurs better at before the onset of disease than when a disease has already occurred. So essentially, throughout life, there may be steps we could take where prevention could be effective, and these steps may depend on the personal circumstances, genetic background, environment. So these are really things that we maybe need to personalize a little bit to obtain the optimum benefit. One of the kind of examples or parallels I like to show is the example of a blood pressure monitor that we all know very well in medicine, and that has transformed, at least in cardiovascular sciences, risk prediction. Because we know that elevated blood pressure is a risk factor, but we can measure it with a very simple tool, and we can then take a preventive action based on risk factors, possibly in combination also with other things. And we can then quantify how effective these measures were. One of the challenges is in aging that we don't have such a simple tool to really measure how the individual aging status is. So what we want, and this is something that I will focus on a lot in my talk today, is biomarkers of aging that could really help us measure and personalize how we approach aging, both in prevention and risk stratification. And obviously, biomarkers of aging alone are good, but not the only thing. So we also need appropriate interventions to really see, can we target processes underlying aging. There are actually quite a number of interesting candidates, and I will introduce some of them. But the question is, are we there yet? So how can we really ensure that these tools can also be translated in the clinic? Let's take a quick step back on really what are biomarkers, because in research and a lot of biomarker developers in the aging context are talking about them, but maybe it's important to see the whole picture. So the FDA treats biomarkers as a quantitative parameter that indicates a biological process or status. So this is a very broad definition, and that is on purpose, because actually a lot of different things can be biomarkers. Examples that are very well known to clinicians will be clinical lab parameters. So even blood values that you might test, so like red cell distribution, neutrophil count, etc. All of these are biomarkers in the end. And many of them are already used. So if in the context of cancer, we may know CA1 to 5 for ovarian cancer, or PSA for prostate cancer. There are many specific disease biomarkers, and this is maybe where they're most commonly used because we have a very specific indication for them. But we maybe want to also have biomarkers of aging, and this is where it becomes a little bit more tricky because, again, aging is a very broad phenomenon. We don't really know when it starts, when it ends, what is aging. Again, I'll come to that in a second. However, such biomarkers should tell us more about our health and our biological age, so kind of how old our bodies are essentially, than chronological age alone, because we know that people that maybe have the same birthday have very different health profiles, depending also on their lifetime history and lifestyles. What could such biomarkers of aging be used for? We're here talking primarily to clinicians. However, there's really a broad range actually. So they could support basic science, looking into aging and rejuvenation, to identify new molecules and processes that target aging. Also for epidemiology, so for research into population aging, identifying maybe risk factors that influence aging. And finally, obviously, clinical research and personalized medicine. This is something that I'm very excited about, to make these tools applicable to individuals and not just on the population as a whole, because we need to make them individually predictive to help translate them into the clinic, and also to see whether certain interventions were effective for a given person before and after. One example that I've been working on quite a lot is epigenetics. And that is because we know that genetics alone plays a big role in how old we become and our disease risk. There's actually almost a bigger part that is influenced by our environment and our lifestyle. So let's think about diet, exercise, smoking, alcohol, infections, also pollution, and of course the aging process itself. And what's exciting is that actually all of these factors become integrated on essentially a single biological process or molecule that's epigenetics, or so-called epigenome, which means nothing more than on top of the genome. My work and that of others has found that epigenetics can record past exposures. So thinking about smoking, viral infections, hormone exposure, or even early life adversity is recorded decades later on the epigenome. And it can also determine present cellular state. So essentially, these epigenetic markers that we have not only capture environmental factors, but they also allow our cells to adapt to them and possibly change their gene expression and behavior. And excitingly, or perhaps often to the detriment, they can modulate future cell plasticity. So how well a cell can adapt to future stimuli and stressors. And they can tell us about future disease risks. So a lot of my work has focused on women's cancers, but we also know from others' work that many age-related diseases and mortality can be predicted using epigenetic signatures. I wanted to give a few examples of what we can use epigenetics for, and I will focus specifically on DNA methylation. That is essentially the addition of these methyl groups to directly on the DNA that we can read out in very high throughput methodologies and very standardized now. So a lot of my work, as I've already mentioned, we can use DNA methylation to detect cancer. We've done a lot of work on cervicovaginal sample DNA methylation and how that would tell us about women's cancer risk. And we found that we can detect or predict breast cancer, endometrial cancer, ovarian cancer, and even cervical cancer from cervical samples that are already used in clinical screening pathways, making it essentially an easy add-on that we could possibly pursue in the future for clinical implementation. I just wanted to give an example from cancer detection because that is something that has now actually made it to the clinic, which we're very proud of because it doesn't always happen in research. So essentially, sorry, this is very small, but we looked at DNA methylation in cervical samples in women without and with endometrial cancer. And we found that there are several regions that have very distinct methylation profiles, so much that we could actually make it into quite simple methylation-specific PCR tests that can now be applied in many different clinical settings because it doesn't need specialized equipment. We validated this initially in a range of settings, including a kind of cohort that had self-sampled cervical samples, ultrasound, and a new test for DNA mutation. And we found that our test actually outperformed clinical gold standard, which is ultrasound, in detecting women with endometrial cancer. And in a biobank sample set, we found that it could detect the cancers before the current diagnosis, so it may enable a much earlier detection. We then worked together with clinicians and scientists at University College Hospital in London to perform a prospective study where we essentially included all women that were referred with suspected endometrial cancer and asked them to provide a sample before the routine pathway. And we then compared the essentially true and false positive rates in those cases. So on the top hand panel, we see out of 100 women, how many would be true and false positive with the essentially ultrasound, which is currently what is used in the clinic. And these women with kind of a red outline are false positives. So you can see out of 100, we have over 50% women that are false positive on ultrasound, which would mean that they are referred, they don't have a cancer, and they then have to undergo biopsies, which are obviously invasive and also a drain on resources in countries like the UK, which has a very constricted NHS at the moment. On the bottom, we have our test, the WIT QEC stands for Women's Cancer Risk Identification Quantitative Endometrial Cancer Test. And we found in the same group, without any further diagnostics, the test correctly identified essentially true negatives and true positives, and had a much lower false positive rate. So that was quite encouraging, and this test has now received approval from the UK Medicines and Health Technology Regulation Association, and we're essentially implementing this, showing that actually the discoveries that we make in the lab can make it to the clinic if we go through appropriate validation. We've also done some work on breast cancer detection and prediction, and this is all published, so I don't want to spend too much time. However, what was interesting is that actually when we look at methylation patterns in different sample types, we find that with blood samples, we are not able to build good predictive algorithms for breast cancer. However, with cervical samples and oral samples, including cheek swabs, the prediction accuracy becomes a lot better. And we think this has something to do with the underlying methylation patterns, which I'm happy to talk about a little bit more. So what else can we detect? We can detect essentially imprints of smoking, and we can also identify certain changes that are shared between e-cigarette users and smokers compared to never smokers, which warrants further investigation of the long-term health risks of these devices. And for instance, based on a saliva or cheek swab sample, we can tell with over 90% accuracy if someone ever smoked, currently smoked, or uses e-cigarettes. So this is quite a lot of information that you have in a single sample. Yeah, I've already mentioned, so some e-cigarettes changes are similar to those in smokers. I've already mentioned we can tell whether someone has had or currently has viral infections, and also hormone influences the epigenome. So those are again published. Happy to talk about those later. And of course, biological age, because aging does change the epigenome, and we can then perhaps use epigenetics to infer how old someone's body is compared to their birthday. So there's a couple of different algorithms that I wanted to point out, because there's a lot of different epigenetic clocks, as they're called, and I wanted to just point out how they're developed. So first generation clocks are typically trained on chronological age data, meaning that we would give DNA methylation data and ask the machine learning model to build a predictor of chronological age. And we're then currently interpreting that any of those where essentially the values are lower than expected, they're aging slower, or they're aging faster. But it's not always clear that this is the best interpretation. So actually, since then, there have been a couple of attempts to make these algorithms a bit more mechanistically grounded. And these are called, for instance, second generation clocks that are trained to predict, for instance, time to event data. So trying to predict how soon someone might die, or predict some more clinically relevant clocks. And then finally, we also have third generation clocks that are looking maybe more into causality or the rate of aging using longitudinal data. So as I mentioned, there's a lot of different clocks, and you may have heard of some of them already. There's not a single epigenetic clock, so they vary by training method, underlying population, and many other factors. This is an example for epigenetics, but the same is also true for other essentially omics. So if you're thinking about proteomics or metabolomics, many of these clocks are trained in the same way, and we're still working towards essentially improving their interpretation. One thing that I wanted to mention in the context of epigenetics, because it's not always thought about, is that actually the same genome makes a huge diversity of different cell types in our bodies. And we often see as epigenetics kind of just a single integrator, but actually a lot of these different cell types are established exactly through epigenetic patterns, meaning that these cells already have pre-existing patterns that may influence them. So one of the things that my work has looked a lot at is how actually all of these factors may interact with different cell types to establish different patterns, because typically a lot of the work is done using blood methylation, and I've already shown that for some diseases this might not be always appropriate. And one kind of cautionary tale or interesting finding I wanted to share is that in a recent study we looked at aging of different or aging patterns in different conditions. So in this diagram, essentially each box would be one aging feature. It's not so important what they are because most of them show quite consistent findings. Anything that's red is kind of higher than in controls, and anything blue lower in controls, and the stars indicate significance. And when we look at breast tissue from breast cancer patients, this shows accelerated biological age compared to control tissue, which was as expected because it's often been described that cancer tissue shows these accelerated aging phenotypes. However, when we look at cervical samples from women with breast cancer, they actually show a decelerated age in some of these clocks. And in blood, there were generally fewer changes. So actually this mimics quite well what we saw in another study, but it was quite surprising to us that we saw this effect, because typically a lot of the studies really showed that cancer patients have accelerated aging, no matter what. But I think it matters a bit where you're looking, and also maybe what these features are telling us. Maybe we don't quite understand. I've also shown that, for instance, smoking and vaping have distinct impacts on different cell types. So if we look at epithelial cells across the body, they show kind of this loss of methylation in genes related to detoxification, which makes sense because obviously these cells are all exposed to toxicants. But in immune cells, we see different methylation changes. Those were related to developmental genes, and then those sites in the directly exposed oral epithelium were showing a gain of methylation in cancer-related pathways. And if we essentially just jumbled all of these cell types together, we would maybe not see these effects. So maybe it's worth looking at different cell types and how they're responding to exposures. Also, one thing to note, we actually find that there are certain age-related changes in methylation that are either shared across lineages. So here we were just looking at epithelial and immune cell lineages as kind of very crude way. There's many more cell types you can look at, but there are a certain number, of course, that are shared, and this is why epigenetic clocks work in general. But then there are some sites that essentially only change methylation or change variability in epithelial cells or in immune cells. And maybe those could also tell us a bit about how different cell types are aging or what's happening there. So this is something that I'm very curious to explore in the future. So I've talked a lot about biomarkers and given some examples, but I hope I've also shown that actually there's a lot of stuff happening and maybe we don't quite know how to go forward to really bring those into the clinic. So as part of the Biomarkers of Aging Consortium, our mission is to establish reliable biomarkers of aging for longevity interventions. And we want to do this by advancing consensus and standards and also collaboration and open science. We were founded in 2022, and we've been fortunate to have the support of many of the field's leading figures in our steering committee, as well as advisory board. And we were very fortunate that Professor Bischof joined us earlier this year. So welcome again. But really, this is a very big community effort to come together and say, okay, what do we need to prioritize? What do we need to do next? So one of the things that we are doing, as has already been mentioned, is that we host a conference every year. The last 2 years we've been at Harvard Medical School, and this is a group picture. So this is actually a few weeks ago. And this was a really great opportunity for biomarker developers, but also people applying these clocks and clinicians to come together to learn about them. So community is essentially the first and maybe most important pillar of what we're doing. But there's also other things. I've already mentioned open science. We need to promote essentially standards across really sharing tools. And one of the things that we've been building is our a library called BioLearn. I appreciate that the majority of attendees will be clinicians, so this is maybe less relevant, but essentially the idea is that we have a very harmonized curation of different biomarkers and clocks that are very easy to apply. And we've worked with the original developers of these clocks to ensure that essentially the result is correct, because there's a lot of different tweaks that you may need to do. So we've already done the work for you. So if you have data, then it's makes it very easy to apply. And we're always also keen to add new clocks and models to our library. We were also hosting a challenge for biomarkers of aging, where we provide prizes for the most accurate predictors of chronological age and mortality. And we're now moving towards our final phase, which will be for health span prediction. So we're still working on some details for that, but that is coming up in the next year. So keep an eye out for that if that's of interest. We also want to collect our own data. And one of the things that we realize is that there's actually a lot of existing data already, but it's maybe sometimes behind very restricted access or very hard to get. So we were like, okay, can we set up maybe a proof of concept longevity study, make it very lightweight for the organizational part, and collect data that could then be collected, over multiple years, and could serve as a blueprint for decentralized studies in the future, where also longevity clinics and other institutes could contribute. So at our conference last year, we had a very big effort to put together resources and volunteers, etc., to host this longevity study. We had 3 days, and it was essentially just from 7 till 9 a.m. before the conference started. We collected blood samples and functional data from 99 participants. So I think it was quite a big effort. Anyone who's worked in trials know how challenging this can be. So huge congrats to the team. So we're currently actually planning a scale up for decentralized sister studies. If you're interested, please do get in touch. We have protocols and materials already available online. So for anyone that is interested in setting this up, please have a look, and also feel free to reach out. And obviously anyone who's interested in partnering or maybe supporting, we're very open for business in that case. And the final thing is that we're also looking into not only building a community, but leveraging that to build consensus and advance the field in that case. Some of the questions that we've tackled in the past were essentially what biomarker can be used when, do biomarkers of aging work, how can we validate them, how can we implement them, and what challenges must be overcome in order for that to happen. So most of the work here has already been published, so I just wanted to signpost you. We had a paper in Cell a few years ago that was looking into terminology and classification of these biomarkers. We also had a paper, I think it was last year or 2 years ago, looking at validation efforts and really what are the next steps that are needed for that. And more recently, we've been working with a very talented clinician, Luisa Georgiana Baca. I don't know, some of you may know her, that has really looked at how we can harness biological age for refined clinical prediction. I think this will be a very exciting manuscript for the community. We're also working with X Prize Foundation currently on how to select and maybe improve immune aging biomarkers for their clinical trials, which I think is a very exciting topic. Lots of buzz around immunity and aging, so very exciting about that. And we've also been working with the Longevity Biotechnology Association on how to harmonize biomarker data collection in clinical trials by early stage biotech companies. And I wanted to give a bit more of an intro to that because I think that might also be applicable to attendees in this crowd. So this manuscript is currently under revision. We hope this will be out very soon so we can share it with all of you. And essentially, we had a focus on what to collect, how to collect it, and why it matters in such clinical trials. We have built a biological sample prioritization framework looking at feasibility, representation, and range of use. And we have a table where we essentially look at different biospecimen and how easy it is to collect, or maybe what they could be used for. And obviously, some samples may be extremely exciting for certain purposes, but less broad. And that needs to be obviously considered for each study. But we have some prioritization. Based on that, our key recommendation were to collect blood samples at regular intervals from trial participants and to store appropriately. So it's important, I think, to have longitudinal data rather than just 1 or 2 time points, because that can tell you a lot about what's happening. And we're also excited about the potential for wearables that can maybe provide more fine-grained information. If possible, even though a trial may only be for a certain indication or safety, it might be helpful to track a wide range of conditions to link those later to outcomes if you might want to study a broader range of topics, and importantly, ensure consent for future uses of samples. So any samples that are collected could be stored and reused for research in the future, because actually what I've come across is that the analyses are expensive, but really the bottleneck is typically samples and data that you have historically. So this would be really a key thing to do. I think this is less important for this crowd, but essentially, obviously, having standards helps everyone to get a higher quality and also have interoperability across trials and the potential for larger pooled data sets. Pre-competitive collaboration can help anyone, not just longevity biotech companies, because we hopefully need to reduce duplication. And I think this notion of aligning on tools, even when we're competing on targets, is quite important. So we're very interested in essentially championing some of these standards and possibly also having joint sample and data repositories, not just with technology companies, but also clinics that could help us scale up these efforts so we can maybe do more than each individual initiative or institute can do by expanding how we share and work together. And finally, one of the work packages that we've done recently was to look at challenges and recommendations for how we can translate these biomarkers. And one of the key outcomes currently was, especially for the very molecular biomarkers, is that we currently don't have very clear interventions or actionable insights. So for example, let's say if I take a sample and I have a higher biological age on epigenetics, what do I do with that? So of course, maybe I can say I want to exercise more and eat healthier, but I already know that I should do that no matter what. So is there something more that I can target? And that's exactly what we discussed. So actually having a very clear insight saying, okay, this level is high, so maybe you can take a certain medication would be much more helpful for a clinician. And this is currently quite a big bottleneck. Another thing that I have been quite interested in is, of course, how can we interpret these epigenetic clocks, and also how malleable are they? So there's now a little bit of data emerging into longitudinal samples and epigenetic clocks, but the majority has been done in cross-sectional studies where we have essentially comparison between people with and without disease, but not really over time. And to answer some of these questions, at the University of Innsbruck, I started together with Professor Martin Wietschwenter and a team of over 50 collaborators collaborators only. Obviously, I couldn't include everyone in a slide, but we had a very strong team to start a new intervention study looking at essentially how preventative efforts would impact on the body as a whole, looking at a multitude of different systems. So we recruited 160 women aged 30 to 60, and the idea was to have healthy individuals that have one risk factor at least. So we had two groups. One of them were overweight or obese with a high BMI, or heavy smokers, and we wanted to see if these individuals undergo essentially healthy lifestyle change, what happens. So we asked them to either do smoking cessation or intermittent fasting, having a 16-to-8 protocol. And we then collected essentially any biospecimen that we could think of. We had a really extremely motivated group of participants who came back every 2 months for these sample collections. So really the biggest thanks of all goes to them. So we had really a large amount of samples that we then subjected to a range of analysis, including DNA methylation across 3 tissues, metabolomics across 2, microbiome profiling across 2, and we also had detailed immune profiling. In addition to all of the clinical measurements that we did, including body composition, functional and vascular exams, etc., as well as wearables and the 2 different interventions. We think this is essentially a multi-dimensional data set across time, sample, omics, and interventions that can tell us, first of all, how malleable each individual element is. So can they change and how quickly? We can also look at essentially what happens as individuals undergo these interventions, because, as mentioned, we know usually from a lot of cross-sectional data or studies that look at one of these, but maybe not all of them together. So what happens? And one thing that I'm also quite excited about is looking at cross-omic and cross-system associations. So let's say we find a change in DNA methylation clocks. What does this correspond to? Or is this preceded or maybe leads to something else? So, of course, this is absolutely a huge amount of samples and data that we are working through. But on a very kind of high level overview, we can just look at which features are maybe more variable or more stable, because those could be informative for how we interpret biomarkers. As we can see, actually, on the more stable end of the spectrum, we have typically flow cytometry, so immune population, as well as actually blood methylation biomarkers. So they didn't tend to move that much. Whereas on the opposite end of the spectrum, we had metabolome profiles, in particular saliva and urine, which again makes sense because these are very strongly influenced by what individuals were eating maybe the day before. So that makes sense that might change between visits quite strongly. We also looked at essentially in these 6 months, compared to the expected direction with aging, how much were these features shifting, and were they going in the same or the opposite direction? So there's quite a lot of information in this diagram, but essentially when we look at this bar graph here, comparing the 2 compliance groups, in the individuals with higher compliance, we found more features essentially that were either attenuated or age opposing compared to lower compliance. And we can also look at this in individual features. So many of them are essentially the higher compliant individuals were, the more slowed it was, although it wasn't the case for all of them. And one thing that we noticed is that actually quite a lot of these changes were in immune cell populations, which maybe wasn't completely unexpected, but there was really quite a lot happening with the immune system. So we wanted to look into this in a bit more detail. And because we looked at so many populations, we thought, is there a way we can combine these insights into a single metrics that could tell us more about what's happening in the immune system? So we derived a new measure called immune age. So it's looking at, I think in total it's 16 populations, and similar actually to other epigenetic clocks or other features. we can predict someone's age and then see maybe how does this change over time. The immune age metric did predict the chronological age to some extent. It wasn't as good as, an epigenetic clock, but we didn't really expect that. And we then looked at this metric over the course of the intervention, which showed us that actually, surprisingly, it's quite small here, but the immune age transiently seemed to increase when people started intermittent fasting. And this was counterintuitive because most of the other effects we saw were actually beneficial, but there does seem to be some impact on how the immune system reacts to basically getting time-restricted nutrients. Although this immune age was only inputted on immune cell population numbers, so essentially, for instance, how many CD4 T regulatory cells people had, we also found that it actually corresponded relatively well to how immune cells might respond to stimulation and how much inflammatory stimuli they made. So we think, actually, although this was not the primary point of the study, we have an interesting metric to quantify how inflammatory and how old an immune system is. We can then also look at all sorts of things. I don't want to spend too much time because there's a lot of information, but essentially we can identify axes of molecular variations. So how, for instance, multi-omic factors would correspond to blood values or essentially questionnaire data like autoimmune disease risk, cardiovascular risk, etc. There's a lot of exciting stuff to look at here, and the first two sources of essentially variation were age and smoking in our cohort, which was great because this is exactly what we expected. So that's kind of a nice sanity check to then dig into the maybe more nuanced findings. And one thing that I wanted to mention here is that obviously what I'm really excited about is how we can leverage this longitudinal multi-omic data to find connections and associations between different features. So for instance, how well would certain blood methylation biomarkers correspond with the microbiome or metabolome or other features? And we can then build, so to say, these networks that find different associations. I'm going through all of this quite quickly because it's only a short talk. However, there is a QR code on top of my slide. All of these data are actually in a preprint, so you're very welcome to have a look at this in more detail. And another way we can look at these associations is essentially across different biomarkers, for instance, in blood, cheek, or cervical sample methylation features, we can find out which other features they might be correlated with. And what was interesting is that actually, yes, blood biomarkers had the most kind of total significant associations, but many of them were really in the blood hemogram. So for instance, neutrophil counts or, other cell counts in the blood or other blood relevant tests. Whereas in the other tissues, they seem to correspond with other features. So that actually tells us maybe there's some level of complementarity and different information captured by depending on which biomarker we're looking at with epigenetics. I'll go over this quite quickly. So we just found some correlations that were expected and some that were new. So we're currently following this up with a bit more detail. So this is some of the work. I've gone over this very, very quickly, but we essentially have a very detailed phenotypes for this group of women that allows us to run a lot of studies. But importantly, obviously, this is almost more data than we could ever look at on our own. And we want to make it accessible to other people. We've actually built essentially a data portal that people can explore some of the primary associations and changes. And we're also trying to make the data essentially as accessible as possible while protecting privacy of participants. Very briefly, what am I interested in the future? Obviously, there's still a lot of work to do with our patient data, but I'm also very excited about moving from correlation to mechanisms. So we've already shown that epigenetics and many other biomarkers are great quantifiable measures that could reflect disease risk. But because it's so crucial to modulating health, it could also be a therapeutic and preventive target. And this is something that I'm hoping to study further in my own research group. So to summarize, I think there's really exciting potential of new biomarkers such as epigenetics. I apologize to anyone who doesn't work in epigenetics and maybe works on another modality. I'm not trying to exclude you. I'm just very excited about epigenetics, but there's a lot of other things as well. I think new longitudinal data collections are great because they can, tell us more about the whole story, provide insights into systemic responses, help us improve interpretability. And we know that there are many different centers and institutes and even clinics that are doing such studies. So I think this is actually a very exciting time to look at decentralization and possibly integrate data. I know it's much easier said than done. There's a lot of legal tape around that, but it's hopefully something that could be resolved. And also the Biomarkers of Aging Consortium helps to provide guidance and material for especially sample collections and partner studies. So if you are interested, please visit our website, or happy to discuss a bit more. With that, I'm at the end of my talk. I'd like to thank my colleagues from King's College London, also my former institute, where we did the multi-omic study, and the Biomarkers of Aging Consortium for the great collaboration in past and future. Thank you, and looking forward to your questions.
Thank you so very much. It was absolutely phenomenal, such a wealth of data and information. I will pass over to my colleague, Dr. Fa, to navigate the questions which we received through various channels. But I also want to mention Dr. Fa. Who is she? She's one of the leading longevity and functional medicine physicians in Thailand. She's one of the co-authors of the Thai national chapter of longevity courses, medicine courses, and one of the people who not only bring longevity medicine to public health on a big scale in the country, but also implemented the medical curriculum of longevity into the National University. So, Dr. Fab, thank you so much. Please go ahead.
Thank you so much for your kind introduction. So, Dr. Chiara, your talk was amazing. And actually, I think our time is too short for you. We can continue for days. So we have a few questions from our audience from Dr. Eric. He asked about your investigation regarding the e-cigarette exposure. He mentioned that the e-cigarette profiles are similar to smokers. Did you also look into vaping and nicotine pouch?
Yes, that's a really great point. Actually, let me stop sharing so I can see you. Sorry, it's and the sun is setting in the UK, as you can see. Yes, so that's a great point. So we looked specifically at e-cigarettes and vapes together because actually at the time the study was done so this was a data collection by one of our collaborators who recruited young people who had either never smoked, were current smokers, or e-cigarette used or vapors. But obviously, actually, since then, there's now been an explosion in different devices. And as researchers, we're almost not keeping up. We haven't looked at nicotine pouches as such. However, we did look at a data set from oral snus or snuff tobacco, which is, I think, very popular in Scandinavia and now becoming popular elsewhere. And we did see that some of those profiles were also recapitulated. What's interesting is that, for instance, in the vaping, we saw that the effects on the oral epithelium were similar to smoking, but we didn't see the same changes in the immune kind of fraction. So it seems there's kind of differentiated response. I don't know what that means long term, but it would actually be very interesting to follow up because we're currently lacking really long-term follow-up. But yeah, with regards to the kind of oral tobacco, that there does seem to be an effect, but it may also differentiate between tobacco and nicotine pouches without a lot of the other products that are in there. So yeah.
Very interesting. And relating to that, I think another question is, what about secondhand smokers? have you have any like intention to look into the secondhand
Mm.
smoker exposure and the epigenetic change?
Yeah, I mean, I think there has been done a little bit. Again, primarily this was done in blood just because it's a lot easier, to look at. I would expect that depending on the dose of exposure, if we look at, for instance, the nasal or oral epithelium, maybe we would see
Mm,
some similar changes, but I don't know. And I think there's a lot of really important work to be done.
okay. And then another question, they asked that on the clinical translation point of view, what do you see as the single biggest hurdle in moving a biomarker of aging from a robust research finding into a widely adopted tool in clinical practice? What is the biggest challenge?
Oh, I mean, there's a few. I think the biggest challenge is that we are lacking the kind of conversation with clinicians. So the
Mm,
researchers do their own thing, and that's what we're trained to do, but we don't actually know what clinicians need usually.
mm,
And I think having that conversation and finding a scenario or indication where we could apply those biomarkers would be, to me, the biggest hurdle. Again, I might be wrong because I am a researcher, so I'm sure there's lots of other challenges. But yeah, so if there are clinicians that are interested in exploring this for a robust use, then I think that would be a huge step for the entire field.
I see. So I think it's very nice because actually, if you answer like that, I think then the researcher is actually act as an innovator, and the innovator have experienced same challenge that sometimes the
Yeah,
communication between
absolutely.
innovators and the user, we need more of
Exactly.
that communication. Yes, totally
Absolutely.
understand.
Yes.
Then another question also regarding standardization. You mentioned your role in the Biomarkers of Aging Consortium beyond the technical standardization. How do we address the challenge of different labs using different clocks
Mm,
and generating different results for the same person? So
mm,
how can we
yeah,
standardize that?
that's a good point. So I don't know. I'm a bit torn for these answers because I know actually one thing that is very nice about for instance, let's say epigenetics community is that most people are using the same platform. So those are the methylation arrays. So we're not really dealing with different products. However, there is still variability when we run the same sample twice, and for a usual biomarker to be implemented, there needs to be super strict, QC and like errors of margin. And I feel that hasn't always been done for the technologies that we use yet. And I don't know if that goes hand in hand with the fact that maybe we haven't tried to move it to that space, that it hasn't been done, or the biomarkers haven't been able to move forward because we don't have the, very strict QC. So we are working towards at least standardizing some of the pre-analytical and post-analytical things. So, for instance, standardizing how the sample is maybe collected and stored, so having similar protocols, as well as having standardized analysis pipeline once the data is there, because actually even for that, depending on which pipeline you use, you might get slightly different results. And that's, people that aren't familiar with this might not know. So this is why we actually implemented our Python library that can deal with a lot of these things. I'm sure there needs to be more done. But again, I think this needs to be done as a community. So hopefully we'll get there.
Okay. And then on the tissue specific cities, how much of a limitation is the tissue specificity? Can a biomarker can be measured in the blood reliably inform us about the aging process, for example, like in the brain or
Mhm.
the liver, like for the organ age?
Yeah, I mean, again, this is a very interesting question because I think it's both a limitation but also a strength in a sense, because you could get different sources of information possibly
Mm,
from different tissues. Obviously, Steve Horvath's work has been amazing to find these conserved signals that are shared by essentially all tissues and even species, looking at mammalian species. Um, what I'm interested in is also looking at further, maybe because we know different tissues age differently. There's tissues that divide a lot, like even the hematopoietic system, whereas in the brain there's much less division by neurons. Yet they share some of these signals, but yeah, we don't know. To my knowledge, for epigenetics, there's been a lot less on looking at organ-specific signatures, although I think there have been some exciting papers recently. I think we need to, yeah, do more research on how well blood can reflect aging in different organs.
That's amazing. And then on one, I think, quite challenging question is regarding the commercial test, about a commercial direct-to-consumer test. There are several direct-to-consumer epigenetic tests are like right now widely available. And in your opinion, what are the biggest ethical and practical concerns you have about the public access to these tests without clinical guidance?
Mm. I mean, I think there's, I don't think any of the companies have bad intentions because they're trying to empower people. And I think it's actually quite exciting that we now have tools to measure. But on the other hand, there is some limitation of what they can tell us because people might overly rely on the results and not maybe necessarily consider other things. I think there is a push towards seeing them in a bigger context, so seeing the person's underlying health status and include that. But again, as you mentioned, without clinical guidance, that's a bit of a problem. I think currently we don't have the validated clinical guidance that they should be used for everyday medicine. But again, if people want to optimize their health, I think it's fair and people should be able to do what they want in that sense.
And then another question, this one go back to your research regarding the smoking, and then you have intervention like either smoking cessation or intermittent fasting. The question is, why do you select intermittent fasting as an intervention to compare with the smoking cessations? Why not other stuff?
No, absolutely. So that is a very valid point. So we essentially wanted to look at two. And they weren't really comparison arms, they were more parallel arms. So we just wanted to identify people with a risk factor and then see what happens. We know smoking is a huge risk factor. So we're like, okay, smoking cessation is one aspect. And on the other hand, diet is a risk factor. And we were actually looking at a variety of different things, including, ketogenic diet and other approaches. And we found that intermittent fasting is generally the easiest to implement because we're not necessarily telling people that much what to eat or how many calories, but really restricting the time. Of course, we're still giving advice to promote healthy eating, but that was kind of the easiest. But yeah, they weren't necessarily comparison arms, which, it would have been very difficult to do.
Okay. And then for the last questions, because I know that you have limited time, for the last question is on the future, looking towards the future.
Yes.
In the next 5 to 10 years, what is the most realistic routine use of epigenetic biomarkers you hope to see in a primary care or preventive medicine setting?
Yeah, so I think there's a lot of exciting potential to measure past kind of exposures and risk factors and taking those into consideration for future disease risk, as well as maybe disease-specific or at least system-specific biomarkers. And this is something that I'm quite interested in. So can we identify which maybe organ system might be the most vulnerable in the person? Because that could also help us improve actionability. And I think those might have an easier way to the clinic. I know everyone wants to move to purely preventive and neuroscience, making it a very broad topic of studying aging. But the reality, and I'm sure you know this much better than I do, is that we're very much system and disease specific. So maybe bridging that gap step by step could be a more easy approach of these biomarkers to the clinic. And then eventually we could make it a bit broader.
Amazing. Dr. Evelyne, do you have anything that you would like to add or
No,
thank you, Dr. Chiara.
Yes, just really want to thank you, especially also for this, what you mentioned in the, at the end. It's very important to really make this connection between sick care and healthy longevity. I also don't think that one can exist without the other, or that we should fuel each other and grow. With this, really, I want to thank you so very much. It was so amazingly great. The recording of the webinar will be posted on YouTube for everybody. And great. Thank you so much. Warmest regards, and see you at the next webinar.
Thank you.
Thank
All the
you.
best. Bye.
Bye-bye.
Thank you.